Hello, my name is Jack DeCruz, and I'm a seventh grader at Menlo Park Academy. My scientific question what is the best product to clean your hands? I chose this topic because in my house, there was a bottle of antibacterial soap that was replaced with bar soap, and I wanted to know which removed more bacteria from my hands. Abstract. What is the best product to clean hands? This experiment tested various cleaning products against bacteria found on a kitchen sponge and a shoe sole. The hypothesis was that soft soap, an antibacterial liquid soap containing benzalkonium chloride, is the best cleaning product because soft soap kills bacteria and it washes away any remaining bacteria on your hand. In the experiment, a thoroughly cleaned hand washed with all of the cleaning products was contaminated with a dirty kitchen sponge or a shoe sole. The contaminated hand was then washed with either Dove bar soap, Purell hand sanitizer, or soft soap, following the CDC guidelines. It was then placed into a petri dish containing nutrient agar and placed in an incubator to promote growth. Every 12 hours, results were recorded and photos were taken. The hand contaminated with a dirty kitchen sponge was best cleaned by soft soap with 237 colonies, followed by Dove with 582 colonies, Purell with too numerous to count colonies, or TNTC, and the hand contaminated but not washed had TNTC colonies as well. In the case of the hand contaminated by the shoe sole, Purell performed the best, with only 4 colonies. It was followed by soft soap with 92 colonies and Dove with 217 colonies. The hand contaminated but not washed had 324 colonies. The hypothesis that soft soap would clean best was supported with the sponge results that showed the least amount of bacterial growth. On the other hand, with the shoe soles, Purell was the best, suggesting that the ethyl alcohol in Purell is better against bacteria on shoe soles than the sponge. Retesting the Purell would be desirable to check for consistency. Scientific question and hypothesis. Scientific question. What is the best product to clean hands? And my hypothesis was, if Dove, Purell, and soft soap each clean a hand contaminated by a dirty kitchen sponge or a shoe sole, soft soap, an antibacterial liquid soap, will remove the most bacteria from the hand because it both kills bacteria and uses water to wash away remaining bacteria. Here's my material list. For the experiment, I used 10 petri dishes, antibacterial soft soap, Dove bar soap, antibacterial hand sanitizer, Purell, an incubator at 80 to 90 degrees Fahrenheit, tap water that was at 90 degrees Fahrenheit, gloves, a dirty shoe, a dirty kitchen sponge, an opaque box that will hold 10 petri dishes and fit inside the incubator, tape to make labels, rubbing alcohol for disposal, and camera to record colonies. If you wish to view different colonies under a microscope, you can use a sterilized toothpick, a microscope slide, distilled water, candle lighter, methylene blue stain, a bowl to clean off the stain, microscope camera, and a computer to see the images. Procedure. For preparation. First, each of the 10 petri dishes will be labeled with white tape. The labels will be clean hand control, sponge no wash, sponge washed with Dove, sponge washed with Purell, sponge washed with soft soap, shoe no wash, shoe washed with Dove, shoe washed with Purell, shoe washed with soft soap, and the petri dish and agar control. To make the clean hand control, wash your hands with Dove and tap water for 90 degrees Fahrenheit for 40 seconds. Next, wash your hands with 1 pump of soft soap and tap water for 40 seconds. After a 1-minute air dry, apply 2 pumps of Purell hand sanitizer. Put your index and middle fingers in the center of the petri dish labeled clean hand control and gently spread your fingers toward the outside of the dish for 3 seconds. For sponge no wash, you wash your hands using the same method as the control. Next, you hold a wet kitchen sponge in your hand for 5 seconds. Perform a light squeeze to get some liquid out and hold for 5 more seconds. Lastly, place your index and middle finger on the Petri dish labeled sponge no wash and spread your fingers like before. For the sponge washed with Dove, soft soap, and Purell, wash your hands the same way as in the control, contaminate with the sponge the same way, and wash with either Dove, soft soap, or Purell for 20 seconds. Place your index and middle finger into the corresponding Petri dishes and spread your fingers toward the outside for 3 seconds, like before. For the shoe sole, for Petri dishes 6 through 9, the shoe sole, use the same process by cleaning your hands with the control. Next, instead of getting your hands dirty with the sponge, hold the sole of a wet shoe for 15 seconds. Follow the same procedure for washing the dirty hand with no soap, Dove, soft soap, and Purell. Put your index and middle finger on the Petri dish the same way as before. The agar and Petri dish control, do not do anything, and label it agar and Petri dish control. Incubation. Put all 10 Petri dishes in a dark box and put the dark box inside an incubator with a temperature of 80 to 90 degrees Fahrenheit. Record data. Every 12 hours, record data by taking photographs of the Petri dishes. Post-experiment. After the experiment has concluded, sterilize all of the Petri dishes by filling them with rubbing alcohol and put the Petri dishes in a Ziploc bag and throw them into the trash. Hand contaminated with sponge, raw data. As you can see in this, sponge that was not hand contaminated but not washed had colonies too numerous to count from hour 12 onward. Purell did the next worst, and after 24 hours onward, you could not count the amount of colonies. Dove was next with 370, Dove was next, and the soft soap was the best. You may notice how as the time goes on, the number of colonies decrease. This is because the area of the colonies grows over time and they eventually merge on the Petri dish and cannot be counted individually. Here's the data for the hand contaminated with the shoe sole. In this, no soap did the worst, as expected. Dove did the next worst. Soft soap did just a bit better than that, and Purell did the best, showing only a maximum of 4 colonies. There are also the controls only had a maximum of 2 in both. And the same goes for before. The colonies on the Petri dish merge, and sometimes the numbers go down after time. And so here's my graph of the colonies from the sponge contamination. As you can see, the black line on top is the sponge that was the hand contaminated but not washed. The brown line represents the Purell, which was also too numerous to count. Next, in the purple line, there is the sponge that was washed with Dove, and the blue line is the sponge washed with soft soap. And right at the bottom, right near the 0 line, you'll notice the 2 controls. Here's the graph from the colonies on the hand contaminated with the shoe sole. Here, the black line is the shoe sole no wash. Next, in orange, is washed with Dove. In blue is washed with soft soap, and right at the bottom is Purell and the controls. And here are my Petri dish images after 72 hours. So these are from the sponge. As you can see, these are the Petri dishes that the data was used, that the data came from. All the way on the left, there is the hand wash with Dove, hand wash with soft soap, hand wash with Purell, and hand that was not washed. And here are the Petri dish images from the shoe sole contamination after 72 hours. All the way on the left is Dove, soft soap, Purell, and the hand that was not washed. Types of bacteria found on the sponge and shoe sole grown on the nutrient agar Petri dish. I was curious at what types of bacteria were growing on my Petri dishes, so to find out what types of bacteria I was growing, I took a bacterial smear to view under a microscope. To do this, I took a sterilized toothpick and took a small sample of some desired colonies and placed them on a drop of water on a microscope slide. Next, I fixed it by moving the slide over a candle lighter 3 times. After letting it cool down and dry off, I dropped methylene blue stain, and after waiting a minute, I dipped it in distilled water to remove any excess stain. As expected, I found both spherical and rod-shaped bacteria, Cocci and bacilli. After doing research, I found that some common bacteria on kitchen sponges and the sole of shoes included Salmonella, E. coli, and Staphylococcus. These bacteria are prokaryotic, single-celled organisms that multiply through binary fission and belong to kingdom Eubacteria. As expected, I also saw some mold on the agar plates. The mold I saw was multicellular eukaryotic organism belonging to kingdom fungi. And here are the pictures I took. These are 2 different colonies I found on the sponge. So on the left is a white colony I found, and on the right is a yellow mold colony I found. And here are some microscope images of some bacteria I found on the shoe sole agar plates. On the left is the orange colony, and on the right was a white colony I found. How the cleaning products work. Dove bar soap. Dove bar soap has no active antibacterial ingredients. Dove works with tap water to wash the bacteria on your hands down the sink. Purell antibacterial hand sanitizer. Purell hand sanitizer contains 65% ethyl alcohol, an active antibacterial agent. Ethyl alcohol is an amphiphil chemical compound, meaning it loves water and fats, both of which make up a prokaryotic bacterial cell's membrane. The ethyl alcohol bonds to the cell's protective membrane and is able to break it. When this happens, the organelles and genetic material are able to spill out and are no longer able to function, and the cell quickly dies. Soft soap antibacterial liquid soap. The active ingredient in soft soap is benzalkonium chloride, an active antibacterial agent that attracts negatively charged bacterial cell membranes because it has a positive charge, and is able to break them open, letting out all of the organelles and genetic material, and therefore killing it. Conclusion. The goal of this experiment was to find the best type of cleaning product for eliminating bacteria on your hand. The hypothesis was that the antibacterial liquid soap soft soap would remove the most amount of bacteria from a contaminated hand because it kills bacteria and uses water to wash away remaining bacteria. The hypothesis that antibacterial soft soap would eliminate the most bacteria was supported with the sponge because it showed the least amount of bacterial growth. With the sole of the shoe, the hypothesis was not supported because Purell had the least amount of bacteria. This may suggest that the ethyl alcohol in Purell is more effective against bacteria found on the sole of shoes. This experiment was successful because the bacterial levels could be observed and the bacterial levels varied in a predictable manner. Additional testing on this subject could include retesting the Purell to check for consistency. This information can also benefit society by informing others of the best cleaning products to avoid common illnesses caused by bacteria. Thank you for listening. I hope you learned something about different hand cleaning products. Thank you.